So today we are going to speak about the central nervous system. And here is a mid-sagittal or a midline view of what we discuss about the central nervous system. We already spoke about the spinal cord, so you see that the spinal cord continues into the brain stem, and then you have the thalamus, and you have the midline view of the one hemisphere. So here we are eventually going to cover everything, but a picture like that can be a nice way to start and a nice way to end. And your goal here is to try to know all the structures that are represented here to a certain extent. Note that you can still add to the labels that are here if you label the lobes or the cortices. So you see the frontal cortex, the parietal cortex, the occipital cortex, and you see the temporal cortex there. The classification of the nervous system based on the embryology is relatively helpful, but not always. So, but regardless of that, let's just start to cover that aspect. So the central nervous system includes the spinal cord and the brain. The brain is called the encephalon. Embryologically, there are three portions of the encephalon: the forebrain, the midbrain, and the hindbrain. And that's pretty much where we could end, but as far as this embryological classification. But note that the hindbrain consists of the medulla, the pons, and cerebellum. Then the midbrain, it's its own part. And then you have these structures called the diencephalon. The diencephalon is perhaps of all these structures relatively useful because you will speak about an area that includes the thalamus and the hypothalamus. And the thing maybe it's our idea of the brain. So we measure the lobes. We think there's the cortex, includes the basal ganglia, and the limbic system is called the telencephalon. So to make my point about the usefulness of it is that you will hear these words and you have to have at least to have an idea of what they mean. So the telencephalon includes the cortex, the basal ganglia, and the limbic. The diencephalon includes the thalamus and the hypothalamus, and I would say that the midbrain and the hindbrain are less helpful because we rather group the brainstem together and the cerebellum apart, and then we have the spinal cord. So I have these videos of a dissection of the sheep brain. You can very well watch other dissections online on YouTube. I have a problem in terms of sound because when I play these videos, the sound gets very distorted. So you use it as you see fit. I think the videos have a caption, so you may even mute the video while you watch it.
Here's the sheep brain with the meninges still attached. The meninges consist of three membranous layers that surround the brain and spinal cord. The inner layer is the pia mater, the middle layer is the arachnoid, and the thick white encasement seen here is the outermost layer, called the dura mater. To remove the meninges, I like to cut around these ventral structures because otherwise they're easily pulled off with the meninges. at the dorsal surface of the sheep brain, we can see the cerebrum. The cerebrum is covered with convolutions, elevations, called gyri. Separated by shallow depressions called sulci. The cerebrum is divided in half by the longitudinal fissure. Into the right cerebral hemisphere and the left cerebral hemisphere. The cerebral hemispheres make up over 80% of the brain. The activities of the right and left cerebral hemispheres are specialized for different functions, making them unique from each other. The cerebrum is separated from the cerebellum by the transverse fissure. As I mentioned, this is the cerebellum. The cerebellum is involved in motor coordination and balance. The surface of the cerebellum has narrow folds called the folia cerebelli. This is the spinal cord. Looking into the transverse fissure, we can look right on the dorsal surface of the midbrain, and you'll notice 4 bumps. This is known as the corpora quadrigemina. It's made up of 2 superior colliculi and the 2 inferior colliculi. If we look just above the corpora quadrigemina, we see the pineal body. The pineal body is part of the diencephalon.
Okay, so this video, again, I'm not entirely sure how the sound is playing, but you can always mute it. The videos do not all overlap exactly with the slides, so the video gave you a lot of details. All the details that are in these videos, we eventually will know throughout the length of this lecture, so they are a nice revision. So let's start with the cerebrum. So the cerebrum is a telencephalic structure following the embryological structure. So we can also call it telencephalon. So as you saw in the video, there are 2 hemispheres, one left and one right. We already talked about that, the distinction between a cortex and the white matter. So the cerebral cortex is the outermost area, which represents that the brain has normally outer gray matter, which is the cortex, and the white matter inside. You see in a picture like that, which comes from imaging, from an MRI, you see the same thing we saw in the video as well. We see that we have something called a gyrus, which is the raised portion, the mountain, if you will, and the furrows, each one of them is a sulcus. The plural of a gyrus is gyri. The plural of a sulcus is sulci or sulci. Let's analyze now more anatomical structures. So the 2 hemispheres are separated from one another by the longitudinal fissure. In addition, 2 hemispheres, each one of the hemispheres is divided by main divisions, which are main sulcus or sulci or sulci. So we have the central sulcus, the lateral sulcus. And these are very important. But there's also another one that's called the parieto-occipital sulcus, which you're not asked to identify, but rather to know that they exist. So these are markings very clear that we can use to divide the hemisphere into lobes. Lobes are portion of the brain. They include gray matter and white matter. So you oftentimes will hear people speak about the cortex, for example, the frontal cortex and the frontal lobe. And you may ask yourself, what is the difference between a frontal lobe and a frontal cortex? And that has to do with the fact that the frontal lobe includes both the white and the gray matter, whereas the frontal cortex only includes the gray matter. So there's another word there, that's the insula, but we are going to see the insula in a little bit, so we can leave it for that slide. So here I ask you to identify the longitudinal fissure and identify the central sulcus. Those are the two main components here. So you have one left hemisphere, one right hemisphere separated by the longitudinal fissure, and here you see the beginning of the central sulcus on each side. You don't need to worry about the parieto-occipital sulcus, which is better observed in a different view. Transitioning to a frontal view, so an anterior view, you see that you still see the longitudinal fissure, but now you see the frontal lobe. Remember the frontal bone, and so see that the frontal lobe takes the shape that fits with the frontal bone. So the space beneath, inferior, has the bone, but we know that below the frontal bone we have the eyeballs on each side. So you see the frontal lobes, so the most anterior portion of the brain, and underneath, and you also see, but perhaps is not the best view, the temporal lobes. And you see the separation from the frontal and the temporal being made by something called the lateral sulcus. This is not the best view, and we are going to surely show you. I have more schematics and pictures that are more illustrative of this, but I like these pictures because these are actual human brain pictures, so they will give a more realistic view. In a posterior view, you see the occipital lobes, and underneath occipital lobes, you see the cerebellum. Note that the cerebellum has also hemispheres. So you have a right cerebral hemisphere, cerebrum, but the cerebellum has cerebellar hemispheres. You keep seeing here the longitudinal fissure, you see the occipital lobes, and relatively more anterior you have the parietal lobes. And so these may be relatively difficult pictures to begin with, but you can go back and forth. But this one is relatively simpler, so let's look at it. So you see here a lateral view and with colors. So you see here one hemisphere, and you see the central sulcus, and you see the lateral sulcus. So our idea for anatomy is we are not going to go very deep as far as function, but we still would like you to know, in a basic course like anatomy, some functions, some very basic functions that we know of some of the lobes. And this image has a very good summary. So I would say if you know all the functions written on the slide in each of those squares, you're actually very prepared for the kind of anatomy we're going for. But before the functions, I would like to walk you through the anatomy. So you see the central sulcus, the area anterior to the central sulcus is the frontal lobe. So the central sulcus marks a separation between the frontal lobe, which is anterior, and is marked here as pink colors, pink or red or orange, and the posterior portion, which is marked in blue, is called the parietal lobe. Posterior to the parietal lobe, marked in green, we have the occipital lobe. Laterally, we have the temporal lobes. So localize the lobes, pause if you need, and then we can speak about some of the primary functions. But again, a picture like this may be very helpful for you to, as a review, once you go through all the slides and the video. So the parietal and the frontal are separated by the central sulcus. The area anterior to the central sulcus is called the precentral gyrus. Precentral, before the central, is the motor cortex, is called the primary motor cortex. We'll speak in a little bit what primary areas are. But for now, let's just keep it that way. Posterior, in the postcentral gyrus, we have the primary somatosensory cortex. So the cortex that primarily deals with sensation of the body. Somato, body, sensation. The posterior to the parietal, we have the occipital lobe, which has the visual cortices. And then the temporal lobe has several functions, but for the kind of anatomy we are going for, I would like you to memorize that in the superior temporal gyrus, we have the auditory cortex. We would have to push aside the frontal and the parietal and the temporal to find a structure that's called the insula. The insula has several functions as far as emotion and feelings, but is also considered to house the gustatory cortex to process taste. Okay, so pause here as you see fit. This is an image just to help you understand the idea, the separation between the cortex and the cortex, which is gray matter, and the white matter. The gray matter includes the cell bodies and the dendrites, and the white matter includes the axons. Note that the brain also has gray matter inside. So the telencephalon also has gray matter inside, which are the basal ganglia, named there. The picture also labels the corpus callosum, which we'll study in a little bit. But it basically is an interconnection between the left and the right hemisphere. So interconnects left with right. Okay? So as you notice, as you have noticed throughout the semester, if you study a little bit more, even if it's just a lab book, the lectures go more smoothly. So this is particularly important also for here, because there are a lot of names that are interconnected. So we can't really just it's only natural that we'll mention things now, concepts now, that now are vague, but then at the end of lecture they become clear. So if you study before, maybe the clarity is reached earlier. So these are the basic principles of the cerebrum. I wanted to now work with you on a more functional aspect, but yet we almost covered everything about the cortex in that slide. But let's devote a little bit more time to speaking about the cortex. So we are not going to speak much about the microscopic aspects of the cortex, but note that the cortical neurons form up to 6 layers, and that the most prominent of those neurons are called the pyramidal cells. So what you see here is work of earlier anatomists that are studying the shape of the cells, that study the shape of the cells in the hopes of find common characteristics and speak about the function. For many years, people abandoned this kind of studies, more cytologic or cytoarchitectural. But more recently, people with new techniques, scientists start looking at the organization of the cells in the brain, and it regains some interest. The other aspect that I would like to talk with you about is the distinction between primary areas and association areas. So this is a relatively well-established concept about the brain, the idea that some areas are primarily devoted to one single function. This may be simplistic, but it's a definition, whereas other areas combine several functions. So areas that are exclusively, quote unquote, devoted to either motor processes or sensory processes are primary areas. So we can speak about the primary somatosensory cortex, a primary motor cortex, a primary visual cortex, and so on and so forth. Then areas of the brain in which that information is mixed, is integrated, we call those areas association areas. Let's just walk through some of the primary areas, but again, we've spoken about that in that slide with the Collard's brain. So the motor cortex, your first question is where? Don't forget, it's an anatomy course, and the motor cortex, the primary motor cortex, is in the precentral gyrus. So it's located anterior to the central sulcus in the frontal cortex. The second aspect is that if you look at that image, you see the exact representation of the areas of the body in the motor cortex. So that, if you look, you see the face being very big, you see the hand much bigger than, for example, the feet. So the reason that there is this difference in terms of sizes has to do with the degree of motor control. So you just learned the muscles, and you appreciate the degree of motor control we have over the face with so many muscles, as well as over the hand. The third thought is that besides the area size, you also observe that this is the location. So medially we have the legs and the foot, and laterally we have the hand and the face, which means that if a person had a stroke in the face area, in the area of the motor cortex that is responsible for motor control of the face, it would represent that the person would have a motor deficit just to the face. Another primary area well established is the somatosensory cortex. The somatosensory cortex is located in the postcentral gyrus in the parietal cortex. The second aspect is that we have equivalent representation to the motor. We have a sensory homunculus, in which, again, the representation follows this pattern, and it's not reflecting the size of the body. So you see the face and the hand, again, take up much more space than the other parts of the body. This has to do with the degree of sensory information that is processed. So it's related to the number of specialized sensory receptors. So we have more sensory receptors in the hand than we have, for example, in our backs or in our feet. The two last primary areas I would like to talk about is the occipital cortex for visual information, but the occipital cortex will have primary and association areas for vision, and the auditory cortex, which is the temporal lobe. All the rest of the brain is thought to be devoted to several functions and form association areas. Note that here you see the size of association areas comparing different animals, and you see that throughout evolution the association areas are the ones that increase the most when you compare animals that are hierarchically superior in terms of evolution with animals that are a little bit lower in the evolution tree. Okay? A classical association area is the Broca's area. The Broca's area is an area that's called speech center. It's a place that, when lesioned, patients will not be able to produce language. They may still understand the words, what they read, what they are told, but they can't produce. That's called Broca's area. Broca is a French anatomist. An equivalent area, but for understanding, is called the Wernicke's area. Wernicke's area seems to provide understanding. So if a patient has a lesion in the Wernicke's area, a patient will be able to produce language, but the meaning is lost. So people may speak a lot, but they are not understanding what you're saying. So they may speak a lot, but the meaning is lost. In both cases, the language tends to be, the hemisphere that controls to the language tends to be the left, even for people who are left-handed. So this is the cortex. Okay, so for our course of anatomy, everything I said is particularly is simple, and that's what you need to know. Again, I mentioned this in one of the videos, but it's important that you understand that we speak about fewer structures, but we are going to go into more detail about each of the structures. So we spoke about gray matter. Now what about white matter? White matter is essentially the fibers, so we're talking about axons. And so there are three types of fibers: association, commissural, and projection. Let's start with the first one. Association fibers are fibers that allow connections between different areas of one hemisphere. So we have something called arcuate fibers, which are more, so areas that are closer to one another, representing blue, or longitudinal fibers. Longitudinal fibers, which form a fasciculus, so we speak about the longitudinal fasciculi, are fibers that may project areas that are relatively far away from one another. So you see, for example, the tip of the temporal cortex being projecting or connecting with the frontal cortex via this longitudinal fasciculi. But as much as we know that the left hemisphere controls the right part of the body and the right hemisphere controls the left part of the body, there has to be interconnection between these two hemispheres. For that we need the commissural fibers. The commissural fibers are interconnecting fibers. So you see here the anterior commissure in the corpus callosum. I'll show you the corpus callosum in a different slide too, for you to have a better idea what it looks like in a midline or mid-sagittal view. The third type of fibers are fibers that project out of the brain. Those are projection fibers. So these fibers are normally organized into what we call the internal capsule. So the internal capsule are the collection of the ascending and descending fibers. So ascending are sensory fibers, descending are motor fibers, for example. And this is an image of the corpus callosum. In mid-sagittal view, an image of one of the models. In a frontal section, you can also appreciate the corpus callosum connecting left with right, and you can also see the internal capsule, projection fibers, and you can also see the anterior commissure. Okay, we are going to now watch another video. Again, this video may help with certain components and may also add additional components that we'll cover throughout the lecture.
To make a frontal section, what we want to do is make an incision to divide the brain into an anterior half and a posterior half. Taking a look at the frontal section, we can see the gray matter that makes up the cerebral cortex. Gray matter consists mainly of neuron cell bodies. Right underneath, we can see the white matter, which is predominantly myelinated axons. Here's the longitudinal fissure, which divides the right and left cerebral hemispheres. This is the corpus callosum, connecting the right cerebral hemisphere to the left. And right underneath that are the lateral ventricles, which are separated by the membrane, the septum pellucidum. This is the fornix. This is the third ventricle. This region is the thalamus, which is part of the diencephalon. Okay, so another view that we can see, so we saw
different views. This view is also very helpful. We are going to come back to this view when we speak about the basal ganglia, but note that in this view you can see very clear the corpus callosum anteriorly, projecting from left to right and right to left, and you see the projection fibers, and particularly you see there the internal capsule. Pause as you see fit to just see these components. Depending on the kind of studies you are following, pursuing, this image may be very relevant in the sense that you see slices like that in MRI and CT scans, and so you may want to spend a little bit more time here, but for now there's still substantial number of structures we need to cover for you to fully understand everything that is represented in this picture. Just a little word about the split brain studies. So you might have heard, and maybe some of you even have those stickers or those covers that say left brain, right brain, or you might have heard people say, I'm more of a left brain or more of the right brain. Great part of those ideas come from studies in which the corpus callosum of the patients was split. The reason for that surgery has to do with epilepsy. But what those patients allowed was scientists to know when the corpus callosum is cut, so there is no interconnection between the left and the right hemisphere, what each hemisphere can contribute in terms of function. That data has limitations, but the principle that one hemisphere works more for a certain function, or in a better way, perhaps saying that a certain function depends greatly on one hemisphere than the other, is called lateralization. And you see in this picture what the incision to the corpus callosum would look like. And we know certain things that are clear. The most important clinically relevant fact is that language is lateralized to the left hemisphere. There's certain aspects of self-awareness that have to do with, for example, perception of our own deficits when we have a stroke, that seem to be more dependent on the right hemisphere. And then there are other functions that are less clear, but you can read more on the slide and also search online for more of that information. So we talk about the cortex and the white matter, and I told you that there is this gray matter inside of the brain that is called the basal ganglia. So I'm not our goal for a course like that, for a basic anatomy course, is not perhaps to have you identify these structures, but rather to know their names and to have a fair understanding, so a basic understanding of their localization in the brain. So the basal ganglia are represented in a picture like that. Depending on the textbook that you use, some people include more structures on the basal ganglia than others. I have a slide from your textbook, so you can follow more what your textbook says. But here I would like to include only the caudate, which you look in the picture represented in blue. You also, if you look at the picture, you see the shape that it has, and it has a tail, and so caudate stands for tail. And then another one that's called the putamen and the globus pallidus. Depending on the textbook you use, you can include more. And you see, for example, here they call it cerebral nuclei. It's an unusual way of putting it, but you can include here the claustrum, which is that little stripe of gray matter, and the amygdaloid. But again, we are going to focus essentially on the caudate, the putamen, and the globus pallidus. Altogether, these three are called the striatum, or the corpus striatum. The putamen and the globus pallidus are called the lentiform nucleus. Okay. So you see here what they look like. So you have one to the right, one to the left. And so you see again the caudate, and then you have the lentiform, which is the putamen and the pallidus. I'm not including the amygdaloid body here, but it's perfectly reasonable to include the amygdaloid body. And that's as far as we go, but I want to give you a little bit better view of it. And the better view actually seen in this picture. So see that the caudate is medial and anterior, and you have the putamen as being relatively more lateral. So which you can actually appreciate if you look at a different picture in the frontal section. So you see the caudate is more medial, and then the lentiform, the putamen, the pallidus, is relatively more lateral. And the pallidus, it stands for pale. So you look at the representation, and it's a little bit, the color is a little bit less gray, if you will, or a light gray. And coming back to another slice, you see the caudate much more anterior. You see the pallidus and the putamen, and that's the composition of the distribution of the basal ganglia in this view. And later on, we talk about diencephalon. We will speak about the thalamus, and you see the thalamus there. So we spoke about the telencephalon. We said that it was the cortex. We talk about the lobes. We talk about the basal ganglia, and now we can talk about the limbic system. The limbic system is perhaps a little bit outdated when it comes to speaking about it, but it's still very helpful for us to classify a set of structures within the telencephalon that otherwise would be very difficult to classify. Here too, depending on the textbooks, you may include more or fewer structures in this group of the limbic structure, the limbic system. So, for simplicity, I will include only the amygdala, the hypothalamus, and the hippocampus. But we could consider other structures, such as the olfactory bulb and other structures, such as the fornix. Collectively, they are thought to be helpful for emotion and learning and memory. They are relatively old in evolution, but the fact that, which means that they are very highly organized, meaning that the size may be misleading. We look at the size of the amygdala, for example, which is represented in purple there, and we think there is a relatively homogeneous structure. But it's actually a structure, there's different groups, separate groups of cells. In a different view, you see here the limbic system. Here we include an additional structure to those we include in a different slide. That is the cingulate cortex, which is relatively superior to the corpus callosum, represented there as well. In a more thorough view, you can have the list of structures that in this textbook they consider the limbic system. Again, it's not mandatory that you know this for now, but it may be the only time we mention certain structures, such as the cingulate gyrus, the fornix, and the mammillary bodies. We will talk a little bit about the hypothalamus, the amygdala, and the hippocampus. Do not worry about the distinction between hippocampus and perihippocampal gyrus. But the hippocampus is that red area, and around it is the perihippocampal gyrus. Speaking about the amygdaloid body, or amygdala for short, is the same. And I just wanted you to understand how we get to learn some things about the brain with the next two slides. So my goal here is for you to know the classification. I'm not interested in you knowing all these details of the limbic system, but I would like you to know that the amygdala and the hippocampus are part of the limbic system. So the amygdala is this tiny little structure represented in purple there. There is one to the left, one to the right, and it's a relatively old structure, so it's very highly organized. And another aspect is that because it's relatively old, you don't need to study the amygdala in humans. You can study the amygdala in many other species, animal species, and you know that it should play the same role, or at least comparable roles, in both species. And so some of the studies you can do, we talk a little bit about lesions, cutting structures, like the corpus callosum, or having a stroke, but we can also electrically stimulate, so make one area overly active. And so what happens in the amygdala, it seems that the amygdala is particularly important for emotion, but even more so for fear. Among all the possible emotions, it seems to be very important for fear. So if you stimulate one of the nuclei of the amygdala, so one of the group of cells, in this case an image of a cat, the cat becomes very aggressive. But a different area of the amygdala, when stimulated, the cat gets very fearful. So that is not the only data, but there are many more pieces of evidence that support the amygdala role in emotion. The hippocampus seems to be particularly important for memory. The role to what extent it depends, memories depend on the hippocampus, and what kind of memories depend on the hippocampus is still somewhat debatable. But I would like you just to give you another piece of evidence, or how another source of studies. So you look at the right and you have what you call a scatter plot. A plot that measures in the x-axis the gray volume of the hippocampus, and the y-axis the years of experience as a cab driver in London. It's before GPS and all that, and you see that the greater the experience, the greater the volume of the hippocampus. So this is what we call a correlation. So the idea is that the bigger, the more I work, the greater the area that I'm using would work. There are other alternative explanations for it. So my goal here with the limbic system is really for you to have a very basic understanding, but also I'm using it to add a little bit of knowledge as to how we know what we know. So we spoke about lesions, we spoke about electrical stimulation, and now we speak about a correlational study in which we measure the size of an area of the brain with the help of an MRI, and then we correlate it with, for example, the performance at one task. So we talked about the telencephalon, and we can end here, and then we will speak about the diencephalon in a separate video.